Schizophrenia, SZ, is a complex brain disorder characterized by diverse cognitive impairments, abnormalities in brain structure and function, and genetic factors. Traditional diagnostic methods are challenged by the overlap of symptoms with other psychiatric conditions, necessitating advanced systems to improve precision. Existing research has primarily focused on imaging data, such as structural and functional MRI, for SZ diagnosis, with less emphasis on integrating genomic features. The proposed Multi-modal Imaging Genomics Transformer, MIGTrans, attentively integrates genomics with structural and functional imaging data to capture SZ-related neuroanatomical and connectome abnormalities. MIGTrans demonstrated improved SZ classification performance with an accuracy of 86.05%, plus or minus 0.02, offering clear interpretations and identifying significant genomic locations and brain morphological connectivity patterns associated with SZ. Imaging modalities, including structural MRI, SMRI, and functional MRI, fMRI, provide unique insights into different aspects of the disorder. SMRI enables the assessment of brain morphology, while fMRI yields connectome data, capturing brain activity patterns and functional connections. Single nucleotide polymorphisms, SNPs, are key genetic variations in the human genome, pivotal for understanding phenotypic traits and disorders such as SZ. Existing methods for integrating multimodal data include simple concatenation, attentional feature fusion, sparse CCA, graph neural networks, and attentive deep CCA. However, these methods may overlook crucial intermodality relationships or have limitations in capturing complex interrelationships. The proposed MIGTrans addresses these challenges by employing a novel three-way stepwise attentive integration approach, leveraging complementary information across genomics, connectome, and SMRI features to enhance SZ diagnostic accuracy. The integration order, genomics right-pointing arrow connectome right-pointing arrow SMRI, is strategically chosen based on inherent correlations between the modalities. Primary contributions of this research include attentive integration for multimodal imaging genomics, clinical interpretability, and the development of MIGTrans, a deep learning model that combines structural brain imaging and functional connectome data with genomic biomarkers. The Multimodal Imaging Genomics Transformer, MIGTrans, is a novel model designed to integrate genomic and connectome data with structural MRI for the classification of individuals with schizophrenia, SZ, and healthy controls, HCs. This model consists of various modules, including genomic and connectome encoders, a structural MRI encoder, and a fusion transformer for multimodal integration. The genomic encoder employs a neural network function, phi, which takes the genomic descriptor, G, as input and learns nonlinear genomic representations, G. This process involves two dense layers with 2048 and 1536 units, respectively, activated by the Gaussian error linear unit, GELU. Each layer is equipped with layer normalization and dropout mechanisms to ensure robustness and prevent overfitting. The connectome encoder uses another neural network function, psi, which takes the functional connectome, C, as input and learns nonlinear connectome representations, C. This process also involves a dense layer with 1536 units, activated by GELU, followed by layer normalization and dropout mechanisms. The output layer generates latent representations of the connectome, vital for capturing intricate brain connectivity patterns. For morphological representation learning, a pre-trained 3D dense net 121 is used to extract morphological features from gray matter density in SMRI images. These features are then passed to a spatial sequence attention (SSA) module for learning attention across spatial and channel dimensions. The SSA mechanism comprises a 3D convolutional (CONVA 3D) layer, a CONVA LSTM layer, and another CONVA 3D layer, which enhance the feature maps by capturing spatial and channel dependencies within morphological feature maps. The evolution of the cell state, QUART, and hidden state, HT, at each time step t is determined by input gate, IT, for get gate, FT, and output gate, OUT, considering both the current and past feature maps in the input sequence. The model's fusion transformer integrates the learned genomic, connectome, and morphological representations, employing cross-modal multi-head attention to distinguish between individuals with SZ and HCs. This fusion process facilitates the integration of diverse data modalities, enabling a comprehensive understanding of the complex interplay between genetics, brain structure, and function in SZ. Here is a combined summary of pages 5 and 6 of the research paper. The Multi-modal Imaging Genomics Transformer, MIGTrans, is a novel architecture that integrates multimodal data, including structural magnetic resonance imaging, SMRI, functional magnetic resonance imaging, fMRI, and single nucleotide polymorphism, SNP, data, to predict schizophrenia, SZ, and healthy control, HC, labels. The CONVA LSTM module in MIGTrans operates based on fundamental equations 5 to 8, involving convolution and elementwise product, to adaptively capture spatial and channel dependencies within morphological feature maps. The Fusion Transformer module integrates learned genomic and connectome features through cross-modal multi-head attention, XMHA, with 2 heads, emphasizing the fusion of functional connectome from fMRI with non-imaging genomic biomarkers. The XMHA mechanism computes attention scores through a scaled dot product mechanism, facilitating selective focus on relevant interactions. The resulting representation is combined with the original connectome features using layer normalization and elementwise addition. The Genomic Connectome SMRI transfuser module integrates genomic connectome features with SMRI features through XMHA, capturing cross-modal relationships between imaging and non-imaging biomarkers. The attention-weighted representations are fused with the original SMRI features through layer normalization and elementwise addition, ensuring the model adaptively combines information from multimodalities while preserving structural characteristics of the SMRI. The fused representations undergo further vectorized fusion through dense layers, retaining cross-modality features alongside fused representations and improving the model's generalization performance. The feed-forward neural network acts as a classification head, utilizing the fused feature representations to predict target labels. The model is trained end-to-end to minimize the cross-entropy loss using the ADAM optimizer. Experiments are conducted on a subset of the Function Biomedical Informatics Research Network (FBIRN) dataset, containing SMRI, fMRI, and SNP data from 186 participants. The proposed MIGTrans model is evaluated using 5-fold cross-validation, and various hyperparameter values are explored to optimize model performance. The proposed Multi-modal Imaging Genomics Transformer, MIGTrans, is evaluated through quantitative and qualitative experiments. The model's performance is compared with various baselines, including single-modality approaches using attention mechanisms, such as genomic SANET, connectome SANET, and spatial sequence attention network, SSANet. The results show that the connectome alone achieves the best performance among single-modal features with 82.33% accuracy, supporting the clinical finding that connectome is a crucial biomarker for schizophrenia, SZ. However, combining genomic and connectome attentive features yields a higher accuracy of 82.71%. The proposed MIGTrans method demonstrates significant improvement in all metrics for two-way and three-way fusion, outperforming concatenation, attentive feature fusion, AFF, and other baselines. The model's stepwise attentive integration of imaging with genomic biomarkers through cross-modal multi-head attention enables the identification of significant genomic locations, brain connectivity patterns, and spatial regions specific to SZ. The qualitative evaluation provides interpretation and clinical validation of the approach by presenting attention maps of SMRI and important connections in functional connectome. The attention maps highlight primary regions in the frontal and temporal lobes in SZ samples, contrasting with thalamic regions in healthy control, HC, samples. This aligns with clinical observations indicating that individuals with SZ experience the most significant reductions in thalamic volumes and thickness within the frontal and temporal lobes. The default mode network, DMN, and cognitive control network, CON, are revealed as significant interactions for SZ classification. The results emphasize the discriminative potential of the attentive integration mechanisms in MIGTrans. The model successfully pinpoints the top 3 SZ-associated SNPs, consistent with a genome-wide association study. The approach offers a comprehensive method for capturing structural, functional brain abnormalities and genetic factors associated with SZ, surpassing the performance of baseline methods and demonstrating its effectiveness in enhancing clinical decision-making and improving diagnostic accuracy. This paper presents a comprehensive review of the current state of multimodal imaging genomics in schizophrenia research. The authors provide an extensive list of references, highlighting the work of various researchers in the field. The references cover a range of topics, including multimodal vision transformers, graph neural networks, and attentional feature fusion. The authors also discuss the importance of imaging genetics in schizophrenia research, citing studies that have used functional MRI and connectomics analysis to classify schizophrenia patients. They highlight the work of researchers who have used machine learning approaches, such as Gaussian error linear units, GELUS, and attentional feature fusion, to analyze imaging genetics data. The paper also touches on the concept of multimodal deep learning, which involves combining multiple types of data, such as imaging and genomics data, to improve disease classification. The authors reference studies that have used multimodal deep learning approaches to diagnose Alzheimer's disease and schizophrenia. The authors also provide an overview of various datasets and repositories that are relevant to imaging genomics research, including the Function Biomedical Informatics Research Network, FBIRN, Data Repository and the GWAS Catalog. They also discuss the importance of voxel-wise gray matter asymmetries in statistical parametric mapping, SPM, and the use of graph convolutional networks for schizophrenia classification. Furthermore, the authors highlight the work of researchers who have used attention-based models, such as the Hinge Attention Network and the Gated Contextual Transformer Network, for diabetic retinopathy severity grading and multimodal retinal image clinical description generation. Overall, the paper provides a comprehensive review of the current state of multimodal imaging genomics in schizophrenia research, highlighting the importance of machine learning approaches and the need for further research in this field. The authors also provide a detailed overview of various datasets, repositories, and analytical techniques that are relevant to imaging genomics research. Equations and algorithms are not explicitly mentioned in these 2 pages, but the authors do reference various machine learning approaches and analytical techniques that are commonly used in imaging genomics research. The paper assumes a high level of technical expertise in the field and provides a detailed overview of the current state of research in multimodal imaging genomics. The figures, tables, and graphs are not explicitly mentioned in these 2 pages, but the authors do provide a detailed overview of various datasets and repositories that are relevant to imaging genomics research. The paper does not explicitly mention limitations, assumptions, or future work, but the authors do highlight the need for further research in the field of multimodal imaging genomics.